So I want to explain to you the steps of DNA replication, and I'm going to use this diagram that I've also posted on Haiku under week number 14. And I'm going to explain DNA replication in the 9 basic steps that occur. This is a double helix strand of DNA. As you can see, it is winded together on this end, but over here it has opened up. And the enzyme that is responsible for opening up this DNA strand is called helicase. Helicase is an enzyme that can unwind and rewind the DNA strand back up together. So this is step 1 of DNA replication, where helicase unwinds the DNA molecule. And you will notice that the DNA molecule now has an open end, this end, we'll call that the 3' end. And if you follow the strand to this side, you'll see that end is the 5' end. We know the strands are anti-parallel, so this end here will be 3'. And if you follow this all the way down here, you know this side is the 5' end. So after helicase has unwinded the DNA at the replication fork, this is what we call this area, the replication fork, a second step is going to occur. A sliding clamp, which we call a beta clamp, is going to attach behind this helicase enzyme, and I'll draw it in like this, and it is going to help to stabilize the DNA. Since the DNA is busy opening up, it's going to be unstable. So if something can help to stabilize it, it's a good thing. Then a third enzyme is going to be required, and we'll label that step number 3, because whenever you have a double helix uncoiling, there is going to start to be a bunching up that occurs in the back here. Just like when you have a rope and made of 2 filaments and you want to unwind that rope, and you pull at the top, it's going to start bunching up. So there's another enzyme that is going to go and attach here, and that enzyme's name is gyrase. It's also referred to as topoisomerase, and its function is to prevent supercoiling happening here in the DNA molecule. So that is step number 3. We're also going to have here on the replication fork some stabilizing proteins bind, and those are called simply just stabilizing proteins, and they do just that. They are going to stabilize the DNA strand that is exposed. All right, so now that we have this replication fork open, we can look at what's going to happen on this strand. This strand up here is going to be what we refer to as our leading strand, and the one down here is going to be what we refer to as our lagging strand. And I'll explain in a moment what is going to happen there. So I want to point your attention to this end of the leading strand up here. You'll see it runs from 3' to 5', but this is the parent or the template DNA strand up here. And we're going to now show what happens when a new strand is copied. That is the purpose of DNA replication. The new strand is copied on top of this parent strand. So this is going to be step number 4. We're going to have an enzyme here play a role. That enzyme is called Rna Polymerase, and I'm going to make it in blue here. And the function of RNA polymerase is to lay down an RNA primer at the 5' end. So you might ask, well, where is that primer? Let's draw it in here. This is the 5' end. So we're going to draw a little piece of red RNA primer in there. This is 5' because it's anti-parallel to the 3' parent strand over here. And when you have that piece of RNA primer laid down, your main copying enzyme, which is called DNA Polymerase 3, that's Roman numeral 3, is going to start to copy the DNA. And it copies the DNA by doing what we call complementary base pairing. And it always does it in the 5 to 3' direction. So if we indicate that happening, we can now draw an arrow showing that it is going to continue on this, what we call the leading strand. It is going to be a continuous process. It's going to start and it's just going to continue. So on this leading strand, you're going to have what we call continuous replication. Now, let's look for a moment at what's happening here at the bottom, at this lagging strand. This same enzyme, the same blue one I drew up there, RNA polymerase, is going to lay down, again, an RNA primer over here. Why over here? Because remember, this is anti-parallel, and down here we had 5', and this runs to 3, so we know that this is the 5' end over here on this piece of RNA. Now, instead of doing a continuous replication, we see something interesting happening on the lagging strand. You're going to have DNA pol 3 come in, and it is going to lay down some of these nucleotides. Well, let's use a color. Let's draw in DNA polymerase 3 here for you so you can see what I mean. So there is DNA polymerase 3 coming in behind this RNA polymerase, and it is now starting to lay down the nucleotides in a complementary fashion. And then it's going to stop. And then you're going to have RNA polymerase come in again, and it is going to lay down another primer. So again, another piece of red primer, RNA primer, and then pol 3 comes in again, and it starts to copy the DNA. And this process is going to continue. You're going to have RNA polymerase lay down a primer, and then you're going to have DNA polymerase 3 come in and start to complementary base pair until it reaches the end over here of this DNA strand. So this is not a continuous replication that is happening. You can see the difference here. It is discontinuous replication. So let me just tell you, I did put that on the diagram that I posted on Haiku. This RNA polymerase enzyme also has a second name, and that is one that's also used in your textbook. It is also referred to as primase because it's laying down that RNA primer. So when DNA polymerase is now finished bringing in these nucleotides and building a short piece of DNA, we are going to have a next enzyme come in, and I'll write that step up here. That enzyme is DNA polymerase 1. And what DNA polymerase 1 is going to do is it is going to come in, and it is going to, I'm going to use a little triangle. It is going to remove these primers. So I'm going to put the little triangle here above each primer to show you. DNA pol 1 removes the primers. And it does so in a 5 to 3' direction. And after it has done that, you need to proofread these pieces of DNA that have copied. And yet another DNA polymerase is going to come in and do that. And that one is called DNA polymerase 2 or pol 2. And its main function is to proofread the newly copied DNA. It is going to fix mistakes in the copying, but it is not 100% effective. Only 1 in 10 million base pair errors still remain, but as you can see, it's not 100% effective. So when you have these little pieces of DNA, which are referred to, we have a name for that, We call them Okazaki fragments. When you have them here, they need to be joined together. And a special enzyme is going to come in. Let's write that name over here. That enzyme is called ligase. And ligase is going to seal these green Okazaki fragments together. So we can put the enzyme in here if you would like to do that. And I'll underline it in yellow. Ligase is going to join these Okazaki fragments together. We call that process ligation, and what is happening is you are sealing the phosphate sugar backbone of this DNA molecule. So in the end, you're going to have a long piece of DNA. The whole DNA strand is going to copy, but it happened in this discontinuous fashion. It's not the same as on the leading strand. Now, when you're done with all of that, there's another thing that's going to happen. Remember, you have now copied this side, and you've copied this side of the molecule, but it is not a helix. So you have to bring in that same enzyme, helicase, to rewind the DNA after you have copied the DNA. So helicase is going to come in, and it was this enzyme that played a role there at the start. To unwind the DNA, it is going to come back in and rewind these two strands together. Now, because you have a parent strand or a template with a new daughter strand on top, you have this scenario. You have a half old and you have a half new DNA. The old is the parent strand, the new is the daughter strand. And because you have this situation happening, we refer to DNA replication as a semi-conservative replication that is happening. Why semi? Because half of the DNA is conserved. You actually keep half of the old DNA and you make half new DNA. So if you can describe these 9 steps that I have quickly summarized for you, and you can do it in a diagram fashion like this, I think that's the best way to answer an exam question. Of course, if you rather want to write an essay, you can do that. But what I'll do is I'll give you a diagram like what I started with here and in your exam, and you are going to be asked to at least be able to summarize these 9 steps.